Good evening, everyone. Welcome you all to the biology series. So today our talk is on cytoskeleton, and our speaker for today is Dr. Parul Bali, who is currently working as a research associate in Department of Biophysics, PGIMER, Chandigarh. She has completed her PhD from Department of Biophysics, Punjab University, Chandigarh. She is editorial advisory board member of Annals of Neuroscience. Her field of interest is behavioral and ocular neuroscience as well as stem cell biology. Along with her, we'll be joined today by our commentator, Professor James R. Bamburg. He is an emeritus professor of biochemistry and molecular biology at Colorado State University, where he has served as the faculty for over 49 years, about 20 of which he held position as chair and program director of interdisciplinary programs. He started research in neuroscience as a postdoctoral fellow at Stanford University and has focused on the neuronal cytoskeleton since starting of Colorado State University in 1971. To further his research interests, he performed 5 years of sabbatical leaves as visiting professor at Cambridge University Laboratory of Molecular Biology, UK in 1978-79, at King's College Cell Biophysics Unit, London in 1985-86, University of Sydney Children's Medical Research Institute, Australia 1992-93, University of California, San Diego, 1999-2000, and Scripps Research Institute, San Diego, from 2006 to 2007. He has published more than 200 papers, most of them focused on the structure and regulation of the cytoskeleton and its role in cell polarities, neuronal development, and neuronal behavior. He discovered the first member of actin depolymerizing factor, Cofilin family of actin regulatory protein, 40 years ago, and has made seminal contributions to understanding their biochemistry, phosphorylation by cell signaling, and their role in neuronal cell biology. His current research is focused on the role and regulation of these proteins in synaptic function and in synapse loss in neurodegenerative diseases, especially in Alzheimer's disease. So with this introduction, I would like to welcome Dr. Parul Bali and Dr. James Bamburg to start today's session. So over to Dr. Parul Bali.
Thank you, Dr. Lee, for the nice introduction. So is my slide visible to you?
Yes.
Okay, let's begin with the presentation. So today's topic is cytoskeletal. Cytoskeletal is the cell's skeletal. From this picture, we can see that there's a cell membrane and nucleus. And in between that, there is a number of proteins which are forming network. These color structures are known as cytoskeleton. There's one quote, form follows function, which means if there is a form, then there is function. If there is a disruption in their form, then there will be a disruption in the functions. So cytoskeleton is a network of filaments and tubules that extend throughout a cell. This supports the cell, it gives space, shape, organizes, and attached with several organelles, and has role in molecule transport and cell division and cell signaling. So in brief history, in 1903, it was proposed that shape of the cells was determined by a network of tubules, and he termed as cytoskeleton. The concept of a protein mosaic that dynamically coordinated cytoplasmic biochemistry proposed by Rodolph in 1929. It was earlier thought that it was uninteresting gel-like structure, gel-like substance will help in the organelle to stay at a single place. There were further studies which discovered microtubules which were vibrating within the neurons in the brain, and it was suggested that brain waves comes from deeper microtubule vibrations. The discovery demonstrates that the cytoskeleton is not just a gel-like substance, it actually has some purpose. Initially, it was thought that cytoskeletal was exclusively in eukaryotes, but in later discoveries, it was also found in prokaryotics as well. So what are the components of cytoskeletal? They are mainly of three types: microfilaments, intermediate filaments, and microtubules. Microfilaments are also known as actin filaments. These are two intertwined strands of actin. The approximate diameter is of 7 nanometer and the subunit is actin. The basic function of microfilament includes maintaining cell shape, changing in the cell structure, muscle contraction, cytoplasmic streaming, motility, and division. So these microfilaments are composed of globular G protein known as G-actin. It combines to form a filamentous structure known as F-actin. So these two forms are reversible. G-actin can go to F-actin and F-actin can become G-actin. So there's another process related to microfilament, which is known as treadmilling. There is the one pointed end and another is barbed end. There is a molecule, a monomeric unit of G-actin. When attached with the nucleotide, that is ATP, it is going to attach at barbed end. And when ATP hydrolyzes to ADP, there is a detachment of the monomers from the pointed end. So this exchange is very important for various phenomena. When this happens, there is a movement of the F-actin molecules, which is crucial for various cell processes. So there are several types of proteins which are associated with actin filament. These are known as actin binding protein. These different proteins have different function. Some of the proteins are giving rise to the cross-linking and another is giving rise to the network formation, and some are stabilizing, another are destabilizing. There are different actin binding proteins. Actins have a major role in muscle contraction. So in muscle structure, we can see there is a myosin in red color, and there in blue, there is an actin filament. These actin filament interact with the myosin. There's another protein, tropomyosin, which covers the active binding site of the filamentous actin. There's another protein, troponin. When there's a high calcium content inside the cells, there is the exposure of the binding site, which was inhibited by the tropomyosin. So this leads to the binding of the myosin, which is in red color, with the filamentous actin, which is in blue color, and causing contraction of the muscle. So second type of cytoskeletal is intermediate filament. It is fibrous protein supercoiled into thick cables. Diameter is of around 8 to 12 nanometer. These are one of the several different proteins of the keratin family. Main function includes maintaining in cells shapes, anchorage to nucleus and certain other molecules, and formation of nuclear lamella. This is the structure of intermediate filament. There's the N terminus, head structure, central rod domain, and C terminus, which is tail. This filamentous structure form dimer, and these dimer join to form tetramers. These tetramers subunit join to form protofilaments, and protofilaments, when joined together, give rise to the filamentous structure. So it is important for cells against the mechanical stress. It provides scaffolds to attract other proteins, including cytoskeletal ones. Another class of cytoskeletal is the microtubule. Microtubule has two subunits. One is the alpha subunit, another one is the beta subunit. Approximate diameter is of 25 nanometer. There's a microtubule organization. Microtubule is attached with gamma 2 RC. It is the center where microtubule is attached. There is the pointed end and barbed end, where all the process of the treadmilling and extension of the microtubules occur. The nucleotide which is responsible in the process of the microtubule is GTP instead of ATP, which was in the case of microfilaments. So this treadmilling process is important. One is the negative end, from where there's the monomers, the alpha subunit and beta subunit are detaching from the negative subunit, from the negative end, and from the plus end, the monomers are joined to form a filament, a tubule-like structure. So there's a dynamic and instability of the microtubules. When there's attachment and detachment of the subunits, which is attached with the nucleotide, either GDP or GTP. Role of the microtubules. These microtubules are the structure of cilia or flagella. So we are here seeing that how microtubules are arranged into 9+2 fashion. There are 9 microtubule structure, and there is two microtubules in the center. So the difference between all these 3 cytoskeletal proteins are of structure. Microtubule, as we know, is a hollow tube structure, and filamentous, these are the intertwined strands, and intermediate filaments are basically fibrous protein which are supercoiled into thick cables. There's a difference between the diameter. Microtubule has 25 nanometer, microfilament 9 nanometer. Intermediate filament is between 8 to 12 nanometer. And there's a difference in the subunits. Microtubule have alpha beta subunit, alpha beta tubulin. Microfilament have actin subunit, and intermediate filaments have there's a keratin family protein associated. There is also a difference in the main function. So here we see how the cytoskeletal components are arranged inside these cells. So this is the plasma membrane, and inside a cell we can see there are a number of organelles which are present, mitochondria and rough endoplasmic reticulum. In the purple curve we can see there are microfilaments residing, and in green color there are intermediate filaments. And we can see there is a microtubule structure in the blue strands. So it is showing that how microtubule, intermediate filament, and microfilaments are spanning and forming network inside the cell. So the key to cytoskeletal functions are, first is the apical region of the cells. We can see here there are villi, and there is the actin filaments. This provides structural and support. Second is the intracellular support. This intracellular support is provided by blue component, which is a microtubule component. There is endoplasmic reticulum and other organelles. This is the picture of a nerve cell. We can see here at the end there are actin filaments which are present, and here we can see in the extension axon of the neurons, there are some microtubules which are present. Over the microtubules, there are some motor protein which are carrying the cargo either from cell body or from the extensions. Similarly, these microtubules and actin filaments have role in mitotic cell division. So we can see here there are microtubules which are attached, which get attached to the chromosomes. There is a protein known as kinetochore, from where the microtubule attach and pull the chromosome and apart from each other during this cell division. So in the process of endocytosis, there's a role of the cytoskeletal proteins, and we can see here the endocytosis and phagocytosis, that is the engulfment of the molecules. It is mediated by several actin filaments. We can see a red portion is of the actin filaments, which is forming a caveolus-like structure, and this is the foreign particle. With the help of the organization of these F-actin molecules, there is the proper engulfment of the foreign molecules. And it is also functioning in the micropinocytosis. Micropinocytosis means there's a engulfment of the liquid molecules. So this is the diagram which is showing the cell body, and another part is the axon. So we can see here the axon length is approximately 3000 times the diameter of the cell body. So whole of the proteins are synthesizing in the cell body. So that means if the protein needs to travel during synapse formation, it has to travel along all these structures to the end. So path is very long. So there must be a system which assists the movement of the protein from one end to the another end. So this is the detailed structure that how molecules are moving. So inside the neurons, we can see there are microtubule structure. Over the microtubule, there are two protein. One is dynein, another is kinesin. These two are known as motor protein. So these are carrying our organelles from one end to the another end. So dynein is basically moving towards the negative end and kinesin is moving towards the positive ends. So the transportation of either proteins or organelles is done with the help of these motor proteins and over these microtubule structure which are residing inside the neuronal cells. So there's a code that neurons that fire together wire together. So we will see the structure of cytoskeletal present in the neurons. In the soma part, we can see here that there's a microtubule structure, and near the plasma membrane, we can see there's a microfilament structure. And in the axon region, we can see the central part is dominated by the microfilament part, and beneath the plasma membrane, there's a microfilament. So dendritic spine, when there is a transmission of this signal, we have dendrites and axon. These two dendrites and axon, these are the two extensions of the neurons. Single neurons has multiple dendrites, whereas only single axon in most of the cases. So axon communicate with the another part of the cells with the dendrite through synapse formation, and these synapse formation is happening because of these dendritic spines. So these dendritic spines are the protrusions. These are the protrusions from the parent part of the neurons or dendrite. These are there's actin polymerization, and there's a dynamic activity of the cytoskeletal component which is giving these structure. There are different shapes of the dendritic spines. One includes the filopodium, another is thin, stubby, mushroom shaped, and the cup shaped. It depends that how synapse formation is occur, and depending on that, these dendritic spine structures are changing. Here we can see the fluorescent images, and on the left side we can see there's a filopodium structures of the dendritic spine. In the 1 week time point, and 3 week time point, we can see the dendritic spine has got the structure like a stubby structures. So these stubby structures have more presynaptic proteins. During the long-term potentiation, there's first filopodial formation, which forms initial contact with the axon button. And further long-term potentiation, there's a formation of new synapses and formation of post-synaptic density proteins. This will further increase their surface area, and there's an increase in the presynaptic molecules. Increase in the presynaptic molecules, and there's a transmission of the synaptic molecules and the signals. Axonal guidance. This is the another interesting phenomena in the neurons. Axonal guidance, also known as axon pathfinding, is the subfield of neuronal development concerning the process by which neurons can send out axon to reach their target. So axon has to travel from their place to the target place, and the guiding molecules are there which can make a path for the axon to reach their target areas. So we have growth cone structure. Growth cone structure is basically a hand-shaped structure at the apical part of the axon. And these hand-shaped structure growth cones are there to identify the targets and also to make contact with the target dendrites. These growth cones are made up of F-actin bundles, and there are the network of the F-actins. This basically have filopodia structure. Filopodia is a finger-like structure, and lamellopodia. Lamellopodia is like web-like hand structure. So we can see in the growth cones that there is a stable microtubule structure. We have motor molecules. These motor molecules are transporting the cargoes from one place to the another place. There is another type of microtubules. These are the dynamic microtubules, means they are moving type of microtubules, and we can see there's a filamentous actin or microfilaments all over the growth cones. So these microfilaments are responsible for the formation of the growth cones, and these growth cones will have receptor which will bind to the, which will identify the molecules from the target areas. So target areas can be the another region of the brain, and it can be the outside the brain. Synaptic plasticity is the another phenomenon. It is the brain's ability to learn and store memory, is understood in terms of change in synaptic connection between neurons. In long-term potentiation, repetitive presynaptic stimulation increases postsynaptic sensitivity and strengthens synapses. This is one of the hypotheses. As we know that memory is attributed to strengthening of the synaptic connection, meaning we say if there's a strong synaptic connection, there is a strong memory. But yet synaptic membrane, we know there's a synapse are the transient structures, and we also know that memories can endure. So this suggests there are some physical basis of the memory. So this study suggests that microtubules, it offers robust information processing with large potential of memory storage. So microtubules can be one of the physical basis of storing the memory. So we now know the basic information about the cytoskeletal. Now, what is the significance of the cytoskeletal? These cytoskeletal, when have some abnormality, give rise to various neurological diseases. For example, there are many studies which indicated cytoskeletal abnormalities in the Parkinson's disease. Alzheimer disease is very common. So in this case, one of the pathologies known as tau pathology. Tau is the molecules which attaches to the microtubule component and stabilizes the microtubule. When there's a mutation in the tau molecule, it won't be able to stabilize the microtubule structure, and there's a destabilization, which causes the development of tau pathology, and there's a memory loss associated with it, and there is neuronal cell death. So these aberration in the cytoskeletal associated protein or cytoskeletal component will lead to the various diseases. Similarly, there's a major role of the cytoskeletal component in the cancer as well. Cytoskeletal is responsible for the metastasis of the cancer cells. So there are many studies that how cytoskeletals is working in various diseases, whether neurological or non-neurological. So in application part, there are various drugs which are being tested for, which are targeting the cytoskeletal component to stop the tumorigenesis or metastasis. Similarly, there are various other therapies for the Alzheimer disease as well as Parkinson's disease at preclinical stage. So number of studies which are targeting are showing some good outcomes. So cytoskeletal holds very important for the study in neurological area as well. So I would like to thank Dr. Axonal for giving me the opportunity to present and give the presentation on this topic. And now I request Dr. Bamburg to share some of their experiences with the cytoskeletal, as he is an expert in this field. Thank you very much.
You can share your screen, sir.
Am I sharing my screen properly? Can you see it?
Yes, yes, we can see it.
Okay. Well, thank you very much for the invitation to participate in this seminar series. And thank you, Parul, for a very nice presentation of a lot of very broad information about the cytoskeleton. So what I plan to do in my talk is to focus in mostly on the dynamics of the cytoskeleton in normal cells and in disease. And I think one way to start this off is to just take a look at a movie of some embryonic tissue. This is a lump of chick pericardium taken from about a 5 or 6 day embryonic chick. And I want you to, excuse me, I want you to watch this video. There are about 10,000 cells in this little piece of tissue. And it doesn't look like they're very active, but when it settles down and attaches to the plastic, you can see dramatic movement of cells. And this dramatic movement is actually taking place within the tissue as well. It's just almost impossible to see it in any kind of three-dimensional structure. I want you to focus in also on that little yellow circle I have there, because you're going to see cell division taking place. Did you all see that cell divide? And there are actually 20 or 30 different cells that are dividing over the time course of this. And if you notice, when the cells divide, they actually migrate in opposite directions from the plane of the division. And this all has to do with the dynamics of the cytoskeleton. If you take a lump of tissue from anywhere in your body and put it onto a culture medium, you won't see this kind of thing, because we have only a very few cell types that maintain active motility and migration in adulthood. But the one case where we actually see this type of development and motility of cells is during tumorigenesis. During this process, cells accumulate mutations and undergo a reversion from a differentiated phenotype to more like these embryonic cells, and they acquire the ability to replicate. But even that is not terribly dangerous, and we can have tumors growing, but as long as they stay within the basement membrane, these tumors are not invading anything. They do not become metastatic, and benign tumors are generally removed with very little difficulty or any kind of long-term consequences. But these cells eventually will accumulate the mechanisms needed to escape from this basement membrane. And these are the kind of processes that are occurring in these very mobile chick embryo cells that I showed you. And these cells can intercalate and force their way through the surrounding basement membrane of a tumor, and they follow along collagen fibrils down to capillaries, where they can squeeze through into the bloodstream and then take up residence elsewhere and become metastatic tumor cells. So if we understand more about the mechanisms that are involved in normal cell growth, we can extrapolate a lot of these to what happens in tumor cells. So this is one of those chick cardiac fibroblasts that I showed you in that first movie. And it's one that was treated with a drug that caused the depolymerization of the actin filament network. And this cell is now repolarizing. So it grows along, it has that kite-like tail behind it that gets snapped in when we throw in the actin depolymerizing drug. Now the cell has a pretty symmetrical lamellipodium all the way around, and it starts collapsing here. So we actually generate the tail of the cell before it actually now starts moving off. And so the polarization actually starts at the rear of the cell, and a collapse of the actin filament network around that rear. And then the ruffling lamellipodia at the leading edge has delivery of material that allows this to take off. And this process of cell polarization is necessary for this kind of polarized migration. This is about a 1-hour movie that's been repeated. Now, I got interested in motility way back in the early 1970s when I was watching neuronal growth cones, which behave much like these cells, and wondering how they actually regulated their actin filaments. And that led me to Cambridge University in the Laboratory of Molecular Biology, where I discovered a protein that I called actin depolymerizing factor, because when you added it to actin filaments, the protein would actually cause the filaments to depolymerize. Another protein was isolated about 5 years later that had very, very similar structures, but when you added it to actin filaments, it bound and sedimented with actin filaments. So it was called Cofilin for being cofilamentous with actin. Well, it turned out that the differences in behavior of cofilin and ADF were very minor. Both could bind to actin filaments, both could depolymerize actin filaments, but they were regulated by pH. And so cofilin and ADF at low pHs both tend to bind to actin filaments. By low pHs, I mean about 7 to 7.2. And if you go up above about 7.4, both of them end up being greater depolymerizing agents. But they both can bind along ADP subunits in an actin filament. So Dr. Bali told you about actin assembling with ATP actin at one end, and then the ATP undergoes hydrolysis, and then ADP actin ends up at the other end, or what we call the pointed end of an actin filament, and the subunits come off. And so they recycle. And when these subunits come off, they have to exchange ATP for ADP, and then they become active in adding on to the end of the filament again. And so this is a steady state that we reach in actin polymerization, but it isn't an equilibrium. So the ends they have different equilibrium constants at the two ends, and subunits treadmill. Cofilin will lead to the severing of these filaments, which generate more ends. And since all ends of actin are where the dynamics are occurring, the more ends you have, the more dynamic the filaments. So some of these severed filaments can nucleate new growth, and some of the severed filaments will cause a more rapid depolymerization. And it depends on the nature of the other proteins that are present, which direction this will go, even in the same cell in different regions of the cell. This shows you where cofilin, or in this case, I think it was ADF, accumulates in a migrating cell. And you can see it's a fluorescently tagged protein, and you can see it all along the leading edge of the lamellipodium, but it is not accumulated at all at the rear of the cell. So what is it doing out at the leading edge? Well, we did some studies where we could inactivate this protein very rapidly by a photo inactivation process. And when we did so, and we looked at the thickness of the actin filaments out here at the leading edge, as soon as it was inactivated, we got a widening of this array, indicating that the actin filaments that were assembling at the leading edge, these are essentially line scans through this fluorescence over time. And this is right when the light was shown to inactivate the cofilin. You can see the band of actin getting wider and wider. And that shows that one of the roles, at least, of cofilin out here is in the turnover of the actin at the leading edge of the cell. One of the model systems that has become really popular in the early to mid 90s to study motility was this intracellular bacterium, Listeria monocytogenes, which gets into the cell. The bacterium itself is non-motile, but it penetrates the cell, and it uses the cell's actin machinery to propel itself around the cell. And in many cases, it will bind to the membrane of one cell and force itself into a neighboring cell, undergo endocytosis into that cell. It escapes from the surrounding membranes, and then it becomes replicates in the other cell. So we can see this comet tail behind these bacteria, and this comet tail is formed from actin. The green here is the bacteria, the red is the actin, and it's assembling due to proteins in the tail of the bacterium that activate the ARP2/3 complex, which causes branching and nucleation of actin filaments. And it's the assembly of actin, no motor proteins are required here. It's the assembly of actin subunits adding in that pushes the bacterium forward in the cell. And then we see what's called array treadmilling as the filaments in the tail depolymerize. Marie-France Carlier was one of the first people to actually reconstitute this using purified proteins and putting onto a piece of fiberglass the protein very similar in structure to what's found in the tail of that Listerium. And so this in an in vitro system, just under a cover slip, would propel these pieces of fiberglass in a forward direction, much like the leading edge of a cell membrane is pushed forward just by actin assembly. There's no epigenetic mechanisms going on here, no phosphorylation, dephosphorylation, other than actin hydrolyzing ATP and being assembled in a way that allows it to push forward on this filament. But you can also see that we don't have a balance here. We're generating a huge amount of actin behind it, and we're not turning it over at the proper rate. So it isn't a good model, but it is a model here. More recently, just in 2020, there was a report from Blecher et al. On a system that they put together where they took beads that they could anchor either an ARP23 activator that would activate ARP23 and allow actin filaments to grow and actually push on this bead. But if we threw in Cofilin, or if they threw in Cofilin here, it would disassemble the system entirely. So they discovered that if they then added formins, which are proteins that nucleate actin filament growth, they kind of walk along the growing end of an actin filament, and they attach these along with the ARP23 at the beads, we'd get these long actin filament structures in addition to the branched filaments from ARP23 complex. And then when they threw in Cofilin, the Cofilin actually would get, a lot of the Cofilin would bind to these actin filaments. They would sever the filaments and depolymerize and keep providing the pool of subunits you needed to keep this process going. If you threw in too much Cofilin, it would cause the disassembly and stop the process of motility of these beads. But just the right amount of Cofilin could be titrated in, and we got this self-regulating system with the cytoskeleton going. And this is just a little movie from their paper showing these beads, and the Cofilin is what's fluorescent here. And so this movie starts at about 30 minutes after the Cofilin is added. We kind of reach a steady state in size of the actin, and you can see that these beads are actually spinning due to the forces that are generated. And so this is very similar to what could occur in the leading edge of a cell, but it provides a steady state mechanism that is dependent on ATP, but does not require any other kind of epigenetic modulation for it to occur. And the same thing is true of a lot of other cytoskeletal regulated systems, that the cytoskeleton is self-regulating in a lot of these events, but superimposed on it in cells is an epigenetic system that allows modulation through extracellular signals. This is the microtubule system in a cell. Microtubules start assembling at the centrosome in a cell, and they we're looking at plus tip complexes that are fluorescent, and so it's only labeling the growing end of microtubules as they leave the place where they're being nucleated by that gamma tubulin ring complex. And each one of these centrosomes, the centrosome will divide and become a spindle pole in a mitotic spindle. Out at the edge of the cells, all we're seeing are the tips of the microtubules. When they get out there and they stop growing, they disappear because the tip complex comes off and we can't see them anymore. But if we actually label the tubulin so that we can see these microtubules, you can actually see the process of dynamic instability going on, especially at high magnification at the edge of a cell where these microtubules are growing and shrinking and growing and shrinking. And this is the normal behavior of microtubules in cells, this dynamic instability. And it is very important in the capture of chromosomes during mitosis, but it's also very important in allowing cells to establish cell polarity. So going back to those original chick cardiac fibroblasts that I showed you originally, some of the cells lose their ability to continue to migrate. And these cells have a microtubule network that is not polarized. But the cells that can overcome this and can actually extend and have microtubules that get captured by the actin filaments in an edge of the cell, these cells become polarized and they can start migrating. So microtubules are necessary for the polarized migration, but microtubules are not necessary to maintain that polarized migration that's driven by actin. So this is a cell that had polarized, and then a microtubule depolymerizing drug was added, and the cells were fixed at different times and shown that the microtubules had disappeared, but the cell maintains a leading edge and it continues a forward motility measured by the centroid of the cell moving forward. The leading edge is rather broken up. It doesn't maintain its nice smooth nature that we saw in the other migrating cells. Now, this is why I got interested in the cytoskeleton to start with. This is the growth cone of a neuron, and you can see that it can respond to cues coming in from this pipette. So the pipette could be dispensing attractive guidance cues, or it could be dispensing repulsive guidance cues that would cause the growth cone to go the other way. These are molecules that have been discovered that are involved in development of the nervous system and the wiring of the nervous system. In this case, we're not actually putting in a so-called guidance cue. We're actually infusing a form of cofilin into the system that can cross the membrane of the cell and actually cause turning by modulating the actin filament network in the leading edge of the cell and stabilizing the filopodia that are coming in the direction that causes this turning. And this turning is also dependent on microtubules being able to penetrate into these actin-rich filopodia and deliver the membrane cargo that's necessary for addition of membrane for the growing growth cone. In a nerve cell that's growing, almost all the new membrane is added in the growth cone of these growing neurons. One other thing that I want to show you is the role of cofilin in regulating contractility. I already talked about its being able to sever and depolymerize actin filaments, but because it can actually bind along actin filaments, if you have a lot of active cofilin that's binding to filaments, it prevents the myosin motors from binding. So this is a bipolar myosin filament. Actin that are in two different directions. So these filaments have opposite polarity. These heads can pull these actin filaments with respect to each other, and they'll slide in opposite directions. If they're attached at things at the end, it generates a contractile force. And these filaments can anchor at the plasma membrane, and they can anchor at the nucleus of the cell, and they can generate contractile forces on the nucleus. And one thing that we can see very nicely is that these contractile forces, I'll show it to you in this movie. This is only about a 8 or 10 minute movie. Chromatin, it shows up dark here in these cells. This was a dividing cell that is missing ADF and cofilin. The proteins have been silenced. Their expression in the cell has been silenced. And you can see the tremendous contractile forces on the nuclear envelope due to hypercontractility of the actomycin without the cofilin to dampen that. You also see these big bleb structures forming on the surface. And I'll show you that these are also myosin based, because as soon as we add an inhibitor of myosin and contraction, the cells, the membrane becomes smooth. So these cells are also missing ADF and cofilin. And what happens is the cortical actin network undergoes massive contractility, forcing the membrane outward as the cortex is pulled together. And that generates those blebs. And these disappear when we get rid of the myosin. Okay, I think this is about the last thing I wanted to talk about. I assume I still have a few more minutes here. So this has to do with dendritic spines. And dendritic spines are also enriched in cofilin. As you can see, the cofilin here is stained in green. Some of these are long immature spines, and some of them are more mushroom and stubby spines that are associated more with memory and learning. And through a series of studies that we and others have done looking at spine shape changes, there are really two electrophysiologically measured events. One is called long-term depression, in which case the spines kind of shrink down and are harder to activate. And then there's long-term potentiation, in which the spines undergo a morphological enlargement, but also have more receptors inserted and therefore are more sensitive to a similar amount of neurotransmitter that's released from the presynaptic terminus. And these spines are more associated with memory and learning. And so we know that the shrinkage of spines is due to the activation of cofilin that depolymerizes a lot of the actin in the spine, and it shrinks up. And to get long-term potentiation, you need a rapid but transient activation of cofilin, probably to sever some of these filaments to form new nucleation sites for more filament growth, and the spine enlarges. And then you need to have cofilin become rapidly inactivated through phosphorylation. And the phosphocofilin essentially collects in the neck of the spine, and this spine becomes a stable structure that is involved then in long-term memory storage. Okay, so the last thing that I wanted to tell you about is the role of these in dynamics of memory and learning. Has to do with the fact that these cofilin stabilized pieces of actin can sit around for a long time. Once you get severing and the pieces is saturated with cofilin, these pieces don't disassemble unless other proteins are here to help it out. So these cofilin saturated actin filaments can actually collect in cells. And if they do so in neurons when you have reactive oxygen species present, such as happens during aging, you get a large increase in reactive oxygen, these cross-link and bundle into what we call cofilin actin rods. And these rod type of structures can be induced in axons and dendrites of neurons in culture by energy depletion, by oxidative stress, and by many of the proteins that are thought to be involved in neurodegenerative diseases, such as the amyloid beta peptides that are involved in Alzheimer's disease, the HIV associated GP120 protein, an envelope glycoprotein in the human immunodeficiency virus, and that protein leads to AIDS dementia, and also the alpha-synuclein filaments will induce rods, and they all use the same pathway for rod induction in neurons. And if we look at these rods ultrastructurally, when they're starting to form, they're thin needle-like arrays, but they can grow to completely occlude the neurite and block transport. Here's a normal neurite. There's a microtubule running down this neurite. This neurite just has this big bundle of rods in here. It has a rod, a bundle of actin. And if we look at Alzheimer brain, yes, we see a lot of these rods in Alzheimer brain, particularly in the entorhinal cortex, which is the place where memory and learning is first disrupted in the synapses of the entorhinal cortex. Okay, so my summary is that ADF cofilin is the principal actin assembly regulator. There are many other proteins which work with ADF cofilins in cells to modulate assembly. It's necessary for polarized cell migration. We can't polarize cells without it. It's necessary for neurite outgrowth and in pathfinding. I didn't show you the requirements in neurite outgrowth, but if you knock it out, you only get a neuroepithelium in the brain of mice that are missing that protein. And you don't get any neurites growing out. And it can undergo oxidative cross-linking and form bundles in neurons. Okay, so I am done with my presentation and turn it back over to Dr. Bali. or
Thank you, Dr. Parul Bali and Dr. James Bamburg for this beautiful presentation on cytoskeleton. So we have a question from our Facebook listener, Dr. Rahul Tyagi. So he wants to ask question to Dr. Bamburg that if to share his thoughts over the hypothesis of encoding and hand wiring of synaptic information elsewhere, for example, in the microtubule lattices.
So I'm sorry, I kind of missed the.
I'll just read it again. So
Okay.
what are your thoughts over the hypothesis of encoding and hand wiring of synaptic information elsewhere, for example, in microtubule lattices?
I guess I really haven't given that a lot of thought. Certainly there are structures there are cytoskeletal structures that can maintain information because of their organization. Whether or not this provides any kind of long-term storage of information, I don't know. So I guess I'll show my ignorance.
So maybe Dr. Parul can comment on it.
Regarding the physical basis of memory and microtubule structures, I've seen only single studies, and it is just showing one of the possibility that memory is storing in the form of the arrangement of these microtubules and one associated enzyme, that is CAM kinase 2 enzyme via phosphorylation. There is a permutation and the combination of the subunits of microtubule and CAM kinase 2 phosphorylated form. So one of the possibility, it may be or may not be. So we cannot say right now.
Yeah, thank you. So I would also request our panel members to if they have any questions for you both.
I have a general question. This was a very good presentation. Congratulations to Dr. Bamburg and Dr. Parul Bali for setting up the biology series and then bringing up cytoskeleton basics into Parul's presentation, and then very insightful and molecular and detailed biochemical analysis by Dr. Bamburg's 40, 50 years of research studies, replete with beautiful live cell imaging. I'm sure it thrilled a lot of audience and inspired many of our alternative practitioners, policymakers, students who are watching this. I thought on behalf of them, I would ask a very, a question that would come to a layman. My question is directed to Dr. Bamburg. Why do a lot of people, philosophers and I would say out of discipline people ask a question that consciousness may reside in the cytoskeleton? Why, if you have come across that kind of there are commentaries and papers on that. Why do they assign it to cytoskeleton?
So a very good friend and colleague of mine, Dennis Bray, with whom I did a sabbatical, wrote an article called The Mind of the Amoeba. And it talked about the various behaviors of the amoeba that are very similar to what we might ascribe to human behavior in terms of the searching out of food and the pausing and almost tasting of its environment, and then being able to go back to a place that it, and this is a single cell organism, mind you, and yet it seems to be able to almost think if you actually watch it in terms of its behavior. And then, so that leads to this kind of argument: if a single cell can actually be storing information and be able to look like it's reasoning, is that really a thought process? And how do we separate that from human thought and behavior? Obviously, we have more ways of dealing with it. But yes, it's a very interesting philosophical argument about what kind of information storage can actually take place in a cell. A lot of this is simply biochemistry, simply modifying in response to something that has occurred in the cell. You get a modification of the membrane or the membrane-associated cytoskeleton in a part of the cell, and when it's going back, it may be because it has more receptors for whatever it is it's looking for that have accumulated now in a part of the cell that it allows it to migrate in that direction. Or instead of more receptors, it has undergone changes in phosphorylation of subunits of something that changes its behavior. Because we know that, you know, directionality and migration can all be epigenetically modified through kinases and phosphatases that work on various proteins, and many other types of modification as well. So how that translates into neurobiology and brain function, I don't know. I have a tough time grasping how it translates into a single cell.
No, but you gave a lot of insights, and thank you for that. Food for thought. And back to Manjari and Parul.
Sir, I want to ask you a question. So, since you were working in vitro culture model, so if we dissect axon, is it possible to regrow by supplementing it with the growth guiding molecules which are required for the pathfinding and for reaching the target? Is it possible to regenerate somehow these axons?
So is it possible to regenerate the axon? So when you say dissect, you don't mean take the axon off of the neuron. You mean to try to regenerate a neuronal axon to regrow. That's what you're saying, if you severed that axon. And there have been huge amounts of studies, of course, that have looked at this with regard to like spinal cord regeneration and what are the things that prevent CNS neurons from regenerating, whereas peripheral neurons we know will regrow. So you can take axons of peripheral neurons, you can sever them, as many people have done when they've cut themselves, and lose the feeling in the tips of their fingers. And over a period of 6 months to 12 months, all of a sudden they'll regain that because the nerve will regrow at a very, very slow rate, but it regenerates. Whereas in the central nervous system, we don't see that regeneration. So the question is, what are the differences? And people have looked at the inhibition of outgrowth by scarring, by the non-neuronal cells being able to prevent the outgrowth. I think Frank Bradke's lab in Germany has done some wonderful studies looking at re-stimulating outgrowth through cytoskeletal manipulations, and has come up, I think just in the last 2 years, they've published maybe 3 or 4 papers on ways of actually getting regeneration of spinal motor neurons. Whether or not you can get them to regrow across severed regions of the spinal cord and get correct connections to be made, I think is still an open question, although people have gotten some decent recoveries. So there's a lot of plasticity. So these cells, if they get over to where they should be, they may find the right connections and make them. One of the things that we don't really think about is when the nervous system actually forms, you know, the embryo and the fetus are pretty tiny when all these things are growing. So we don't have to grow neurons to go 1 meter in length from our spinal cord to our toes when we're just a little developing embryo. They grow a few centimeters and they've made their final connections. And yet in an adult, there's often a long way to go for regeneration.
So there are some molecules which are activating the formation of neurites. So are these molecules helpful in the enhancement of memory in animal models, if we say?
Well, people have used things like brain-derived growth factor, BDNF, and other growth factors that have been infused, and to try to get regeneration and in animal models, and there has been some success in improvements in cognitive function, but nothing that I think would qualify as a major breakthrough yet. but yes, they're stimulating regeneration, and the use of stem cells in terms of trying to do the same thing. There have been a lot of labs that have been looking at this, and you can get some improvement in cognitive function. We're looking right now at inhibitors of rod formation that actually seem to cause much greater improvement in things like Alzheimer mice in terms of their cognitive abilities. Whether or not it is doing it by blocking rods or having some other effect, we don't know, because the pathway for rod formation is a fairly broad one that involves the prion. It involves prion protein and NADPH oxidase. And if we inhibit those, we can block rod formation.
Thank you, sir. Is there any question from anyone?
While the others
I want to ask one question.
Please go ahead.
My question is whether doing meditation will slow down the process of degeneration.
Whether doing what will slow it down?
Meditation.
That's getting outside my field.
I can
I think my wife thinks it will. And she keeps trying to get me to meditate.
Let me rephrase that question for you, make it more biochemistry oriented. Have there been studied on mice that have been treadmilling in the cage and versus those that haven't been exposed to exercise with the same diet, same environment, and has anyone chopped off their brains and looked at longitudinally the rods formation? It's not that close, but exercise helping you.
No, but so this is very interesting. So we had a seminar just recently from a woman who works on obesity and different types of fat and how these actually affect brain function. And it turns out that people have reported either increases or decreases in brain function depending on obesity, but that obesity has always been measured by the what do they call it? The index for measuring body fat, essentially, weight index. Whereas what she finds is that there really is a strong correlation between subcutaneous fat and visceral fat. And people that have a pear-shaped body with more subcutaneous fat in the hips actually have a lot more of what are called beige fat cells. And those beige fat cells actually produce an anti-inflammatory cytokine that actually has a beneficial effect in the brain. And this is not produced by subcutaneous fat. And so actually they have an inverse, even though they're obese and overweight, they actually have an improvement in their brain function by the anti-inflammatory cytokine. Whereas normal fat will eventually lead to the production of inflammatory cytokines, the IL-1 betas and IL-6 and things like that. So diet, exercise, and where you're storing your fat apparently can have a big effect on brain function. And most of this work was done in mice. So there were obese mice that were being used for these studies, and they did look at the brain, but not at the level of rod formation. They were looking at just brain mass and whether or not there were activated astrocytes in the brain, not rods.
Thank you for a very nice answer to that question. Are there any more questions from Anjali or Parul? Or from the Facebook?
No, sir, not from Facebook. So no one is having a question, then I